Assalamu alaikum, hello guys, welcome back to my channel. Today I wanted to just show you something quickly about some main equipments of microbiology lab. Guys, I will tell you something. If you want to learn about biosafety in laboratory, actually I will not explain this in my channel because I will enter the explanations, enter the details quickly, and I will go to culturing because it will take a long time for me to explain about biosafety. You can go to other videos actually, but I will explain about techniques of culturing, streaking, and of course I will explain about interpretation of the results of cultural results. Okay, what we need in a microanalytic balance, autoclave, Bunsen burners, centrifuge, colony counter is not so much necessary. Actually, we don't have it in our lab, homogenizer or a vortex, microscopic slides, and tube racks. Okay, of course we need a microscope too, that I forgot to write it. Microscope is important for lab, for even a pathology, a microbiology, any type of biological study, mycology, parasitology. We need microscope for all of those. Analytic balance is used for to measure. Okay, autoclave is used to sterilize the substances, liquid substances, and glasswares. Okay, Bunsen burner and swabs, of course. Bunsen burner is a heat. Okay, is just like is a source of heat which creates a sterile environment for culturing, and it prevents contamination. Okay, centrifuge, as you know, all you know what's a centrifuge. Colony counter, deep freezer for storing some cultures. Homogenizer, microscopic slides. Of course, microscopic slides are needed for gram staining and sometimes blood smear. What are the sterilizing equipment that we should have? We should have autoclave and disinfectants. So what are disinfectants? Such as alcohol. We disinfect the surface, okay, by alcohol. Microwave, in some labs it's used, and ovens, but the main two are autoclave and disinfectants. And we have iodine that's used for skin, it's a skin disinfectant, and alcohol, sometimes bleach, is used to sterilize or to disinfect the working area. Okay, safety equipment are needed, of course. Laminar flow hood or biosafety cabinet. What's a biosafety cabinet? It's just like a cabinet which in the upper part contains. Actually, biosafety cabinet is much better than a laminar flow hood, but it's much expensive than a laminar flow hood. So the biosafety cabinet simply pulls the air and filters the air. We can say it has a HEPA filters which filters the air, okay, and prevents contamination of your sample, and it protects you from any infections and or aerosols. So before doing any tests in the biosafety cabinet, first, of course, you disinfect the area. You disinfect the area of the biosafety cabinet that I will show you now. You disinfect it by any disinfectant, good disinfectant, and after that you will turn on the ultraviolet light. So the biosafety cabinet has a light that's called ultraviolet light, which is used to sterilize, even to clean or to kill the bacteria on the surface of the biosafety cabinet. So the when you turn on ultraviolet light, you should be very careful because it's dangerous for you. Don't you look at the light, or don't you put your hands under the light. About maybe 20 minutes, it's enough for me. When I do a culture, I just turn on the light for 20 minutes. After the 20 minutes, actually some people say you can turn on a fan before turning on the UV light, but no, I don't do this step. Actually, I just turn on the UV light and enough. After that, I turn off the UV light and I will do my work. What's my work? Is it culture or pouring plates in it? Bunsen burner, as I said, it's even a safety equipment that's needed. It's used to sterilize wire loops, okay? PPE, personal protective equipment. PPE, such as gloves, masks that are PPE, face shields, those are PPE, okay? Culturing equipments, which are very important. We have media. Guys, let me give a simple explanation. Media comes in many forms. Uh, in their forms, they come in liquid, solid, and semi-solid, okay? The liquid is called broth, okay? And oh my god, we forgot something to write, which is incubator. Okay. Where were we? Yeah, medias. Medias are, you know, like humans, like any other organisms, bacteria need things that they're necessary for their life. Okay. Okay, or you can say growing. They grow by using those things. We have different types of media such as maconchi, blood, okay, nutrient, agar. We have a lot of types. XLD, hecton, enteric. I will explain them when we enter the diagnosis. I will explain about the types of media. Okay. And this flasks and glass tubes. So the flasks and glass tubes are used to prepare the media. So what's a media? It comes in a form just like this one, as you see here. It's written agar listeria. Okay, here as this form. It contains a powder that it's written on it how much weight you should prepare for distilled water. Okay, it's prepared in distilled water and mixed. Okay, and autoclaved. If it's some medias don't need autoclaving. Okay, it's written on it. Depends on the type of the media. You autoclave it and you let it to cool for until 50 degrees Celsius, and you pour it in things that are called petri dishes. It's just like preparing a gelatin, but you need a balance. Okay, you need a balance to prepare it. You need to weight it by grams. Okay, as you see, those are called petri dishes or petri plates. There are two forms of them. The glass one is reusable and you can sterilize it by using autoclave, and the plastic one, which is disposable, you can't use it, reuse it. Okay, glass and glass tubes even are used for preparing some types of media such as triple sugar iron media. Uh, Wire and plastic sterile loops. A loop is something just, I don't know if I brought a picture of a loop. Okay, I didn't bring, but loop is something just like that. I will show you. Okay, the loop, sorry. It's something like that, yeah, but it's smaller than this actually. It comes with an end, which you put it in a liquid, this end, and you do culturing, streaking. Okay, we have two types. We have wire that you use it, you heat it to sterilize it, and we have plastic sterile loops that you use it for one time and you throw it, okay, in a biohazard box. Here is the technique of pouring media. Pouring media should be in biosafety cabinets or what's called near a Bunsen burner. Okay, okay, this is a media. This is a flask that you pour the media in it. You pour it in petri dishes. Here we have a glass petri dish, which can be used so much. It's common for use. This is an autoclave. Okay, autoclave. You should be very careful dealing with autoclave because it has it occurs pressure on the sample and heats the glasswares by stem. Okay, those are important to know. Those are called transfer swabs. Okay, or sterile swab stick. Okay, Those swabs are sterile, means if you touch anything, the bacteria or the microorganism will go on the tip end of this swab, and you put it in the tube of it, okay? This is called a sterile or a transfer swab, and it's sterile. This is a Bunsen burner that I told you. You should work near the Bunsen burner after sterilizing the surface. You should work near a Bunsen burner because to make a sterile area of working and preventing dust and contamination to enter your culture media. So this is the biosafety cabinet. As you see, this microbiologist does a bacteria culture by a loop, by a plastic loop. This is called a loop, which she holds, not the plate, which looks like a dish. No, not the circular thing. This long thing structure. It's called a loop. This is called a loop. Okay, a loop is used for bacteria culturing or streaking. Okay, this is a plastic loop. Okay, plastic sterile loop. This is the biosafety cabinet, because the biosafety cabinet, as you see, she puts her hand in the biosafety cabinet and does her work. You it's better to not use Bunsen burners in biosafety cabinets if you have biosafety cabinets. Okay, so thank you guys. Don't forget like and subscribe. Hope you get benefit.